Concept number 4. The endomembrane system regulates protein traffic and performs metabolic functions. Many of the different membrane-bounded organelles of the eukaryotic cell are part of the endomembrane system, which includes the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, the various kinds of vesicles and vacuoles, and the plasma membrane. This system carries out a variety of tasks in the cell, including synthesis of proteins, transport of proteins into membranes and organelles or out of the cell, metabolism and movement of lipids, and detoxification of poisons. The membranes of this system are related either through direct physical continuity or by the transfer of membrane segments as tiny vesicles. Despite these relationships, the various membranes are not identical in structure and function. Moreover, the thickness, molecular composition, and types of chemical reactions carried out in a given membrane are not fixed, but may be modified several times during the membrane's life. We will now focus on the endoplasmic reticulum and other endomembranes to which the endoplasmic reticulum gives rise. The endoplasmic reticulum biosynthetic factory. The endoplasmic reticulum, ER, is such an extensive network of membranes that it accounts for more than half the total membrane in many eukaryotic cells. The ER consists of a network of membranous tubules and sacs called cisternae, from the Latin cisterna, a reservoir for a liquid. The ER membrane separates the internal compartment of the ER, called the ER lumen, cisternal space, from the cytosol. And because the ER membrane is continuous with the nuclear envelope, the space between the two membranes of the envelope is continuous with the lumen of the ER. Here we have a nice figure of the ER. This is a membranous system of interconnected tubules and flattened sacs called cisterna, and the ER is also continuous with the nuclear envelope. The membrane of the ER, as you can see, encloses a continuous compartment called the ER lumen. Rough ER, which is studied on its outer surface with ribosomes, can be distinguished from smooth ER in the electron micrograph, or the TEM. Transport vesicles bud off from a region of the rough ER called transitional ER and travel to the Golgi apparatus and other destinations. So, in summary, there are two distinct, though connected, regions of the ER that differ in structure and function: smooth ER and rough ER. Smooth ER is so named because its outer surface lacks ribosomes. Rough ER is studded with ribosomes on the outer surface of the membrane, and thus appears rough through the electron microscope. Now, the Golgi apparatus is essentially a shipping and receiving center. After leaving the ER, many transport vesicles travel to the Golgi apparatus. We can think of the Golgi as a warehouse for receiving, sorting, shipping, and even some manufacturing. Here, products of the ER, such as proteins, are modified and stored and then sent to other destinations. Not surprisingly, the Golgi apparatus is especially extensive in cells specialized for secretion. Now, the Golgi apparatus consists of a group of associated flattened membranous sacs called cisterna, looking like a stack of pita bread. A cell may have many hundreds of these stacks. The membrane of each cisterna in a stack separates its internal space from the cytosol. Vesicles concentrated in the vicinity of the Golgi apparatus are engaged in the transfer of material between the parts of the Golgi and other structures. A Golgi stack has a distinct structural directionality, with the membranes of cisterna on opposite sides of the stack differing in thickness and molecular composition. The two sides of a Golgi stack are referred to as the cis face and the trans face. These act respectively as the receiving and shipping departments of the Golgi apparatus. The term cis means on the same side, and the cis face is usually located near the ER. Transport vesicles move material from the ER to the Golgi apparatus. A vesicle that buds from the ER can add its membrane and the contents of its lumen to the cis face by fusing with a Golgi membrane on that side. The transface on the opposite side gives rise to vesicles that pinch off and travel to other sites. Products of the endoplasmic reticulum are usually modified during their transit from the cis region to the trans region of the Golgi apparatus. For example, glycoproteins formed in the ER have their carbohydrates modified, first in the ER itself, then as they pass through the Golgi. The Golgi removes some sugar monomers and substitutes others, producing a large variety of carbohydrates. Membrane phospholipids may also be altered in the Golgi. In addition to finishing its work, the Golgi apparatus also manufactures some macromolecules. Many polysaccharides secreted by cells are Golgi products. For example, pectins and certain other non-cellulose polysaccharides are made in the Golgi of plant cells and then incorporated along with cellulose into the cell wall. Like secretory proteins, non-protein Golgi products that will be secreted depart from the transface of the Golgi inside transport vesicles that eventually fuse with the plasma membrane. So let's have a look at lysozymes and their digestive compartments. A lysozyme is a membranous sac of hydrolytic enzymes that many eukaryotic cells use to digest, hydrolyze macromolecules. Lysosomal enzymes work best in the acidic environment found in lysosomes. If a lysosome breaks open or leaks its contents, the released enzymes are not very active because the cytosol has a near neutral pH. However, excessive leakage from a large number of lysosomes can destroy a cell by self-digestion. Hydrolytic enzymes and lysosomal membrane are made by rough ER and then transferred to the Golgi apparatus for further processing. At least some lysozymes probably arise by budding from the transface of the Golgi apparatus. But how are the proteins of the inner surface of the lysosomal membrane and the digestive enzymes themselves spared from destruction? Apparently, the three-dimensional shape of these proteins protect vulnerable bonds from enzymatic attack. Now, lysosomes carry out intracellular digestion in a variety of circumstances. Amoebas and many other unicellular eukaryotes eat by engulfing smaller organisms or food particles via a process called phagocytosis. The food vacuole formed in this way then fuses with a lysosome whose enzymes digest the food. Digestion products include simple sugars, amino acids and other monomers, and these pass into the cytosol and become nutrients for the cell. Some human cells also carry out phagocytosis. Among them are the macrophages, a type of white blood cell that help defend the body by engulfing and destroying bacteria and other invaders. Food. Well, this picture shows a lysosome fusing with a food vacuole during the process of phagocytosis by a protist. Lysosomes also use the hydrolytic enzymes to recycle the cell's own organic material, a process called autophagy. During autophagy, a damaged organelle or small amount of cytosol becomes surrounded by a double membrane, and a lysosome fuses with the outer membrane of this vesicle. The lysosomal enzymes dismantle their inner membrane with the enclosed material, and the resulting small organic compounds are released to the cytosol for reuse. With the help of lysosomes, the cell continually renews itself. A human liver cell, for example, recycles half of its macromolecules each week. The cells of people with inherited lysosomal storage diseases lack a functioning hydrolytic enzyme normally present in lysosomes. The lysosomes become engorged with indigestible material which begins to interfere with other cellular activities. In Tay-Sachs disease, for example, A lipid digesting enzyme is missing or inactive, and the brain becomes impaired by an accumulation of lipids in the cells. Fortunately, lysosomal storage diseases are rare in the general population. Vacuoles, diverse maintenance compartments. Vacuoles are large vesicles derived from the endoplasmic reticulum and the Golgi apparatus. Thus, vacuoles are an integral part of a cell's endomembrane system. Like all cellular membranes, the vacuole membrane is selective in transporting solutes. As a result, the solution inside a vacuole differs in composition from the cytosol. Vacuoles perform a variety of functions in different kinds of cells. Fluid vacuoles formed by phagocytosis have already been discussed. Many unicellular eukaryotes live in fresh water, have contractile vacuoles that pump excess water out of the cell, thereby maintaining a suitable concentration of ions and molecules inside the cell. In plants and fungi, certain vacuoles carry out enzymatic hydrolysis, a function shared by lysosomes in animal cells. In plants, small vacuoles can hold reserves of important organic compounds, such as the protein stockpiled in the storage cells in seeds. Vacuoles may also help protect the plant against herbivores or by storing compounds that are poisonous or unpalatable to animals. Some plant vacuoles contain pigments such as the red and blue pigments of petals that help attract pollinating insects to flowers. So here we have a plant cell vacuole. Mature plant cells generally contain a large central vacuole, which develops by coalescence of smaller vacuoles. The solution inside the central vacuole, called cell sap, is the plant cell's main repository of inorganic ions, including potassium and chloride. The central vacuole plays a major role in the growth of plant cells, which enlarge as the vacuole absorbs water, enabling the cell to become larger with a minimal investment in new cytoplasm. The cytosol often occupies a thin layer between the central vacuole and the plasma membrane, so the ratio of plasma membrane surface to cytosolic volume is sufficient even for a large plant cell. So let's have a look at the endomembrane system. So here we have at the top left hand side the nuclear envelope, and it's connected to the rough ER, which is also continuous with the smooth ER. And as we move down, we can see that the membranes and proteins produced by the ER move via transport vesicles to the Golgi, and from there the Golgi pinches off transport vesicles and other vesicles that give rise to lysosomes, other types of specialized vesicles and vacuoles. Here we at the bottom we have the lysosome, and this becomes available for fusion with another vesicle for digestion. And then we have the transport vesicle, and this carries proteins to the plasma membrane for secretion. And finally we have the plasma membrane expands by fusion of vesicles. Proteins are secreted from the cell.